In this PowerPoint, we are going to cover the physiology that occurs at a synapse. A synapse is the location of communication between the nervous system and maybe another neuron, or a muscle, or an organ, or a gland. I want you to be familiar with the anatomy that is at a synapse. And this lecture, we're going to actually just focus on synapses between two neurons. So at the distal end of an axon, you have a swelling, and that's called the axon terminal, also known as a synaptic bulb or synaptic knob. And at the most distal aspect of that axon terminal, you have a presynaptic membrane. The space that's between the sending neuron and the receiving neuron is filled with interstitial fluid, and that's called the synaptic cleft. Then the receiving neuron would have what's called the postsynaptic membrane. And within the membranes of both the postsynaptic membrane and the presynaptic membrane, there are ionic transmembrane proteins or transporter proteins within the membrane itself. There's two types of synapses in the nervous system. There's the chemical synapse, which is the most common one, and we will talk most extensively about that one. And the chemical one uses neurotransmitters for communication. The second one is an electrical synapse, and that uses mostly gap junctions where the ions from one synapse will just flow through to the next neuron to stimulate the receiving neuron. There's very few of these in the central nervous system, but they are abundant between smooth muscle and cardiac muscle. We'll talk about those when we get to those subjects. This slide of the oculomotor nerve is just one example of an electrical synapse in the CNS. Location of chemical synapses. Well, you have one that goes from a nerve cell to an organ cell. That example is over on the right, it shows it go into the adrenal gland. And then you have a nerve cell to a muscle cell, that's on the bottom lower right, showing a neuromuscle synapse. And then you have nerve cell to nerve cell, which is on the left side in the bottom. There are different types of neurochemical synapses in the nervous system. You have axodendritic, which is the axon of the sending neuron and the dendrite of the receiving one. You have axosomatic, which is the axon of the sending neuron and the cell body of the receiving neuron. Then you have axonic, which is the axon of the sending neuron communicating right at the axon hillock in this diagram with the receiving neuron. And that happens to be the most common location of motor signal synapses. And then you have a few that are dendrodendritic, but there's not too many of those. Events at a chemical synapse with a stimulatory neurotransmitters. Now, we've covered this a lot in the past, but you will be expected to know when these events occur and what's happening. So when an action potential arrives at the end of the axon terminal, it will open voltage-gated calcium channels, and this will allow an influx of calcium into the axon terminal. The influx of calcium inside the axon terminal will stimulate the process of the vesicles that are filled with neurotransmitters to dock with the presynaptic membrane and eventually release the neurotransmitters into the synaptic cleft via exocytosis. Then that empty vesicle in the presynaptic membrane will reform by endocytosis and then get refilled again with neurotransmitters. There are three types of vesicles found in a neuron: small clear, small dense core, and large dense core. The small clear ones, they contain neurotransmitters such as acetylcholine, glycine, GABA, and glutamate, just to name a few. These small clear vesicles with their neurotransmitters are synthesized and recycled at the axon terminal. The small dense core vesicles, they contain catecholamines such as norepinephrine, epinephrine, dopamine, and these too are synthesized at the axon terminal and recycled there as well. Then you have large dense core vesicles, and these contain neuropeptides. Neuropeptides are large, and so they are synthesized at the cell body, not in the terminal, and they have to be transported to the axon terminal via antegrade axoplasmic transport. Okay, vesicles have what's called snares, and snares is an acronym for soluble N-ethylmalamide sensitive fusion protein attachment protein receptors. I will only use the acronym SNARES from now on, and that would be what I'd use in an exam as well. So I just want to draw your attention to the diagram on the right, and you see there's an example of a vesicle, and you see these kind of purple angled things sticking out of them. And then you see a lighter lavender angled bars or rods coming out of the plasma membrane of the cell. And those are snares. And you have two types of snares. You have the V snares, and the V stands for the vesicle. These are the ones that are on the vesicle. And you have T snares, and these are the ones that are within the are considered transmembrane snares. I talked a little bit about this in the beginning of the course when we covered membrane transport. So I'm just reminding you here that vesicles have snares, and so does the cell membrane of the presynaptic neuron that's going to be releasing the neurotransmitters into the synaptic cleft. The synaptic vesicle cycle is really involved, but it has four main phases. The first phase is considered docking, then you have priming, then next is fusion combined with exocytosis of the neurotransmitter, and then endocytosis. This is a very involved process, and we're going to get into those processes next. Okay, before I can talk about the process of synaptic vesicle cycle, or how a neuron actually releases a neurotransmitter via a vesicle into the system, we've got to cover some terms. So the first term here is calmodulin, and calmodulin is an intracellular regulatory protein. Next, we have an enzyme called CAM kinase, and it is a protein kinase enzyme that phosphorylates ATP. We have RAB3, which is a small g protein, then NSF, which stands for N-ethylmalamide sensitive fusion protein. We have Synapse, which stands for soluble NSF attachment proteins. SNARES, which are SNAP receptors. Synaptotagmin, which is a vesicular membrane protein that has calcium receptors on it. And then you have synaptobrevin, which is a vesicle membrane protein. It's a V snare. And then you have syntaxin and SNAP25, which are plasma membrane proteins. These are the T snares. And then lastly, we have clathrin. Clathrin is a geometric protein that provides the framework for a vesicle to reform by endocytosis. We're going to get into the steps of a vesicle releasing the neurotransmitters. So to reiterate, first an action potential has to arrive at the axon terminal, opening the voltage-gated calcium channels, and you have your first influx of calcium. Once the calcium enters the axon terminal, it will bind with calmodulin. This calcium-calmodulin complex will activate CAM kinase, which will release vesicles from a pool. Neurons have the ability to make extra neurotransmitters and store them in the vesicles within the axon terminal. And they store them on a rod-shaped protein called synapsin. And this area where they have the stored neurotransmitters is referred to as a pool of vesicles. Once the vesicle is released from the pool, energized Rab3 proteins will guide the vesicle to the active zone on the presynaptic membrane. The active zone is an area of the presynaptic membrane with a high concentration of voltage-gated calcium channels. As the depolarization wave wraps around the most distal end of the axon terminal, it will open up the voltage-gated calcium channels at this active zone, allowing in another second influx of calcium. Once the Rab3 has brought the vesicle to the active zone, that is called docking. This docking brings the V snares and T snares in close proximity to each other. Next, the vesicle needs to be primed, which energizes it and energizes the V snares, the synaptobrevin, to intertwine with the T snares, which is syntaxin and SNAP25, and they will wrap around each other. Then the next phase is fusion and exocytosis. Fusion occurs when the second influx of calcium at the active zone binds with synaptotagamin, that is one of the transmembrane proteins on the vesicle, and it will stimulate the synaptobrevin to want to bind with phospholipids in the presynaptic membrane. Well, as it reaches or goes towards to bind with the phospholipid, it kind of wraps around the V snares and the T snares, making them very snug and creates a very tight zippered-like junction. And then it will pull, the snares will pull apart, and as they pull apart, they open the vesicle, causing it to release all of the neurotransmitters into the synaptic cleft. Okay, this slide here just lists all the steps of synaptic vesicular exocytosis in order. I did this in case it seemed kind of choppy going through slide to slide via just studying this on your own. But I'll read to you the order of them. One is the action potential, opens voltage-gated calcium channels at the axon terminal, and you have your first influx of calcium. So the calcium will then enter the presynaptic terminal. The calcium will bind with calmodulin. The calcium-calmodulin combination will activate CAM kinase. The CAM kinase will cause vesicles in the pool to be released. Rab3, energized by GTP to GDP, will guide the vesicle to the active zone, and the vesicle will dock. The dock vesicle is then primed by hydrolyzing ATP. Then V snares, known as synaptobrevin, will intertwine with T snares, syntaxin and SNAP25. An additional voltage-gated calcium channels will then open in the active zone. The calcium will bind with synaptotagamin, causing it to wrap around the snares, both the T snares and the V snares, as it fuses with phospholipids in the presynaptic membrane. Then the snares pull apart and open the vesicle, releasing the neurotransmitter. Calcium effects on neurotransmitter release. The amount and frequency of calcium influx is directly proportional to the amount and frequency of neurotransmitter release. Therefore, if you increase an action potential frequency or its strength, you'll increase the amount of intracellular calcium, which will increase the amount of neurotransmitter release. Action potential voltage drops at the axon terminal. The telodendra, if you remember, are narrow and unmyelinated, which increases resistance of the current going down the axon itself. This will cause the voltage to drop to as low as 4 to 11 millivolts. However, that's still strong enough to open calcium voltage-gated channels. Now, if you have rapid firing, if another action potential quickly follows behind the first action potential, if you have rapid firing, that'll increase the action potential voltage, will increase more calcium channels opening, and thereby increasing neurotransmitter release, which will increase the postsynaptic response. We've learned about how vesicles release the neurotransmitters. Now we're going to learn how the vesicles can refill themselves through endocytosis. The first thing that's going to happen is that the snares need to unzip. And they'll do that by two proteins, one called Synapse and the other NSF. And those two proteins will unzip or untangle the V snares and the T snares that were intertwined. Then there's a geometric protein called catherine that will surround the vesicle, and that'll act as a framework for the vesicle to reform. Then a protein called dynamin will pinch off the vesicle from the presynaptic membrane. The catherine will then fall off, and then the vesicle has two choices. If that axon terminal is getting frequent stimuli, it'll just refill itself, because vesicles actually have on their in their membrane, they have two transport mechanisms. One is a proton pump, and the other is a proton neurotransmitter exchange. So it will use the proton pump to pump in a whole bunch of hydrogen ions, and then it'll do an exchange of hydrogen ions with neurotransmitters that are in the cytoplasm, and it'll fill itself with neurotransmitters. Another option is that the newly formed or recycled vesicle can blend with an endosome, where the endosome has these same pumps on it, a proton pump and a proton neurotransmitter exchange pump. It'll get filled with neurotransmitters at that point, and then it will join with the rest of the vesicles. It'll pinch off and join with the rest of the vesicles to release the neurotransmitters. So that pretty much completes the cycle. Steps written down all at once, so I'll repeat that. Number 1, Synapse and NSF unzip the snares. 2, catherine covers the vesicle. 3, dynamin pinches off the vesicle. 4, catherine falls off once the vesicle is formed. 5, vesicles either fuse with endosomes or they refill themselves. And 6, they use primary and secondary transport mechanisms to refill the vesicle, whether it's doing it on itself or in the endosome, using a proton pump first to fill it up with hydrogen ions, and then using a secondary pump to exchange a proton for a neurotransmitter molecule. Events at a chemical synapse with a stimulatory neurotransmitter continue. So now we've had the neurotransmitters released into the synaptic cleft. Now these neurotransmitters have to diffuse across the cleft via kinetic energy, and they will bind with ligand-gated ion channels that's on the postsynaptic membrane. It takes 2 neurotransmitter molecules to bind with an ion channel to activate it. The ligand channels will open and will allow sodium to enter the cell. Synaptic delay. We're familiar with pathways of the nervous system. We know that the sensory system has a minimum of 3 neurons, first order, second order, and third order, that need to synapse to send the signal all the way to the cortex, and that the motor system has at least 2 neurons, an upper motor and a lower motor, to send the information from the cortex to the receiving tissue. Well, every time you have a synapse, it can slow down the communication process, particularly if these synapses are chemical synapses. So the communication to and from the brain requires several synapses, and it can take a lot of time. It can take 0.3 to 0.5 milliseconds. Doesn't seem like a lot, but that does take time. So the more synapses there are in a neural pathway, the more time it takes to send and receive messages. Excitatory or inhibitory. Synapses are one or the other. They're either excitatory or inhibitory, and it depends on the neurotransmitter being released and the types of receptors on the receiving neuron. Neurons have the capability to receive both excitatory and inhibitory signals simultaneously. They can have up to 1,000 synapses on their bodies, or cell bodies, or axons are all over them, and receive that amount of signals all at once. Dendrite and soma synapses are difficult to propagate an action potential. For one reason, they're unmyelinated. Another is they have few, if any, voltage-gated sodium and potassium ion channels. They do have voltage-gated calcium channels, which will help with neuroplasticity, and we'll talk about that later. So dendrites and soma synapses will require repetitive firing from the presynaptic neuron in order to reach threshold, which is local depolarization, in order to send a signal strong enough to make it to the axon hillock so it can facilitate the action potential. Local depolarization at an excitatory synapse. Excitatory postsynaptic potentials, EPSPs, are local depolarization that move the membrane towards zero. It makes it less negative on the inside, and they do this by opening ligand-gated sodium channels. These local depolarizations vary in size and strength from approximately 0.2 to 5 millivolts, and they last about 15 milliseconds. Fast EPSPs begin depolarizing after about 0.5 milliseconds and will peak between 1 to 1.5 milliseconds. And then they rapidly decline via conduction with decrement. However, they can be summated. And this summation can also bring the soma or the dendrite or wherever the synapse is closer to threshold, as long as the subsequent action potential arrives before it has reached resting membrane potential. So this summation can actually lead to an action potential. Local hyperpolarizations at an inhibitory synapse. Inhibitory postsynaptic potentials are IPSPs, and they move the membrane away from zero, making it more negative on the inside. They do this by opening potassium and chloride channels. They too vary in size and strength. It's almost the same as the EPSPs. Their strength varies from 0.2 to 5 millivolts. They last about 15 milliseconds. The fast IPSPs peak around 1 to 1.5 milliseconds, and they rapidly decline as well. They can be summated too. So a neuron or inhibitory signal, if it's strong enough, can nullify an excitatory signal. On the previous two slides, I talked about fast EPSPs and fast IPSPs. And on this slide, I'm going to give a little bit of information about slow EPSPs and slow IPSPs. And as you can imagine by their name, they last several seconds. They're slow. They occur in the autonomic nervous system ganglia, the cortical neurons, and cardiac and smooth muscle. Slow EPSPs will decrease potassium conduction out of the cell. That will help the inside of the cell stay more positive, moving it closer to threshold. And they do this by affecting voltage-gated potassium channels, keeping them closed longer. Whereas slow IPSPs will increase potassium conduction out of the cell, and they do this by making voltage-gated potassium channels stay open longer, allowing more conduction of the potassium out of the cell and making the inside of the cell more negative and stay more negative longer. There are two ways a neuron can be summated. One is spatial and one is temporal. The spatial will be where two separate neurons, are communicating with one. Whereas the temporal, it'll be one neuron communicating with one neuron, but it's in repetitive firing. Divergent synapses is one to many. One axon will form synapses with several neurons. Therefore, one impulse can stimulate several excitatory postsynaptic potentials. Convergent synapses is many to one. This is where several neurons can form synapses with the same receiving neuron, or one neuron forms several synapses with one receiving neuron. And these several synapses can create several impulses leading to one excitatory postsynaptic potential. Response latency and learning. Response latency is the time span between a stimulus and a response or reaction to the stimulus. Response latency is affected by three things: the length of the axon, conduction velocity, synaptic transmission or delay, and we talked before that a synaptic delay can be as much as 5 milliseconds. Then we have learning. Learning reduces response latency. It does this by increasing summation, which will lead to decreased synaptic delay. This is all about neuroplasticity, which we're going to get into next. Neuroplasticity is the ability of a neuron synapse to grow in size and in number. Mostly it affects cerebral and cerebellar cortical dendrites. The results from repetitive high-frequency action potentials, which are referred to as tentonic stimulation. Repetitive high-frequent action potentials are tentonic stimulations, can lead to synaptic potentiation. And synaptic potentiation is neuroplasticity. Synaptic potentiation is a result of repetitive stimulus of a synapse, rapid firing, also known as tentonic stimulation. It involves the neurotransmitters glutamate and acetylcholine. Synaptic potentiation physiology. When you increase the use of a synapse, that leads to increased presynaptic calcium channels opening on the axon terminal, which will increase the amount of intracellular calcium at the axon terminal, which will increase the amount of neurotransmitters being released. The more neurotransmitters you have released, the stronger the excitatory postsynaptic potential will be. A strong postsynaptic potential will lead to the opening of postsynaptic calcium channels on the receiving dendritic spine. This opening of calcium channels on the dendritic spine will lead to an increase in dendritic intracellular calcium. This is the key to increasing the response to subsequent stimuli and the key to neuroplasticity. There are three types of synaptic potentiation: facilitation, post-tentonic potentiation, and long-term potentiation. Facilitation is just very short-term plasticity. It lasts less than 10 to 100 milliseconds. It does not affect any postsynaptic sensitivity. Post-tentonic potentiation is also short-term plasticity, lasting 1 to 10 minutes, but it does not affect postsynaptic sensitivity. Whereas long-term potentiation is going to have some effect. Long-term potentiation leads to long-term plasticity. This can last for weeks, and if used enough, will last a lifetime. It will increase the postsynaptic sensitivity, meaning it increases the sensitivity of the dendritic spine receiving that signal. It primarily involves glutamate and acetylcholine neurotransmitters. Long-term potentiation can lead to the growth of the dendritic spine and increase the number of dendritic spines on the receiving neuron dendrite, which will increase the number of synapses. This also affects both pre- and postsynaptic neurons. As if you increase the number of dendritic spines, you need to have an increase of axon terminals to communicate with them. And this whole process will involve protein synthesis. You need proteins for structures to grow. How does increased calcium via long-term potentiation cause dendritic spines to grow? Well, the increased calcium within the dendritic spine will bind with calmodulin. This complex will stimulate CAM kinase, a protein kinase, to stimulate protein synthesis. You'll have strands of mRNA that migrate from the soma to the dendrites. The proximal dendrites also have nisobodies which are able to synthesize proteins. The dendritic spine can produce enough proteins, cytoskeleton, protein channels. It can produce whatever it needs to grow that dendritic spine. You will also have an increase of calcium within the axon terminal, which will stimulate that rescinding axon also to grow. Maybe grow even more telodendron to facilitate more synapses on that receiving dendrite, which will lead to neuroplasticity. Dendritic spine pruning. If a dendritic spine can grow, it can also be pruned. It's one of these use it or lose it types of physiology. Pruning can be caused by pathologies or genetics or just aging, but mostly it's of a lack of use. Backwards action potentials. What happens is when an action potential is generated at the axon hillock, sometimes it'll travel backwards towards the dendrites. And this will cause synaptic potentiation of those dendrites that it reaches and causing new dendritic spines to grow. Hippocampus, cerebellum, and long-term potentiation. Long-term potentiation is responsible for short-term memory in the hippocampus. The more you study something, the more you review something, helps your dendritic spines to grow so you can remember the information. The motor memory in the cerebellum is also a part of this process, and the more you practice something, a physical activity, you will have motor learning in the cerebellum. Presynaptic inhibition and facilitation at an axonal synapse. This means two axons synapsing with each other, where one of those axons is also trying to synapse with another dendrite. It's going to be confusing for me to try to relate this just audio. It's better in person, but I'm going to give it a go. So the drawing on the left has a green axon terminal, a yellow axon terminal, and kind of a grayish purple receiving dendrite. So we're going to say that the yellow axon that has an action potential going down it is going to try to release a stimulatory neurotransmitter. Doesn't matter which kind, as long as it's stimulatory. Let's just say acetylcholine. So it has the action potential and it's reached the axon terminal. Now on the left, the green axon terminal is releasing GABA, which is an inhibitory neurotransmitter. Now we already talked about how calcium is necessary to release the vesicles from the pool there so they can dock with the presynaptic membrane and release the neurotransmitters. Well, the GABA is going to inhibit the calcium, voltage-gated calcium channels on the axon terminal, so very few will open. And if the amount of calcium that enters the axon terminal is directly proportional to the amount of neurotransmitter that will be released. So in this situation on the left, the GABA is going to significantly reduce the amount of neurotransmitter that will get released at that synapse at that time. Now on the right side, The green axon terminal is now serotonin. And we're going to just still say that the yellow one is going to release acetylcholine, and you've got a receiving dendrite, and it's in gray. So when the action potential comes down the yellow axon terminal, the serotonin from the green axon terminal will be released, causing more calcium channels on that axon terminal that has the action potential coming down it to open. And that'll allow more calcium to enter the axon terminal. The amount of that calcium within an axon terminal is proportional to the amount of neurotransmitter release, and that will facilitate a lot of neurotransmitter being released at that particular synapse. I really hope this makes some sense. I apologize if it doesn't. Hopefully you'll understand it with the notes as well. So here I try to write the process down. Presynaptic inhibition and facilitation caused by axonal synapses. Inhibition is when an inhibitory axon terminal releases an inhibitory neurotransmitter on an excitatory axon terminal that releases an excitatory neurotransmitter. It will decrease the size of the action potential reaching the terminal, which will decrease the number of voltage-gated calcium channels opening, which will decrease the neurotransmitter released, and that'll decrease the excitatory postsynaptic potential. If it's facilitation, it's an excitatory axon terminal releases an excitatory neurotransmitter on an excitatory axon terminal that releases an excitatory neurotransmitter. This will prolong the action potential and increase its strength, which will lead to more voltage-gated calcium channels opening and staying open longer, which will increase significantly the amount of intracellular calcium at that level, which will increase the amount of neurotransmitters released and increase the excitatory postsynaptic potential. There are other factors that affect a synapse, such as an increase in body temperature. That can cause a synapse or the receiving neuron to be hyperexcitable. And this can lead to febrile seizures. If you've ever witnessed somebody having a little seizure when they have a high temperature. Also, decreased pH can lead to hyperexcitability of a neuron because it lowers the threshold of the receiving neuron. Fatigue of a neuron, if it's overstimulated, it can actually run out of neurotransmitter. Because if it's overstimulating, it's releasing neurotransmitters faster than it can recycle it. And this usually recovers rather quickly in a few seconds on its own, especially if the neuron stops the firing. I hope that this lecture, this PowerPoint makes some sense. If it doesn't, please feel free to contact me. I'll do my best to try to explain it further.